So here we have a typical diagram of a neuron cell, and the ability of a neuron to receive and transmit information is based on highly specialized cellular organization. Most of a neuron's organelles, including its nucleus, are located in the cell body. A typical neuron has numerous highly branched extensions called dendrites. Together with the cell body, the dendrites receive signals from other neurons. A neuron also has a single axon, an extension that transmits signals to other cells. Axons are often much longer than dendrites, and some, such as those that reach from the spinal cord of a giraffe to the muscles in its feet, are over a meter long. The cone-shaped base of an axon, called the axon hillock, is typically where signals that travel down the axon are generated. Near its other end, an axon usually divides into many branches. Each branch end of an axon transmits information to another cell at a junction called a synapse. The part of each axon branch that forms the specialized junction is a synaptic terminal. At most synapses, chemical messengers called neurotransmitters pass information from the transmitting neuron to the receiving cell. In describing a synapse, we refer to the transmitting neuron as the presynaptic cell and the neuron, muscle, or gland cell that receives the signal as the postsynaptic cell.
Potassium ions and sodium ions play an essential role in the formation of the resting potential. These ions each have a concentration gradient across the plasma membrane of a neuron. In most neurons, the concentration of potassium is higher inside the cell, while the concentration of sodium is higher outside. The Na plus and K plus gradients are maintained by the sodium-potassium pump. This pump uses the energy of ATP hydrolysis to actively transport sodium out of the cell and potassium into the cell. The sodium-potassium pump transports 3 sodium out of the cell for every 2 potassium that it transports in. Although this pumping generates a net export of positive charge, the resulting voltage difference is only a few millivolts. Ion channels allow ions to diffuse back and forth across the membrane. As ions diffuse through channels, they carry with them units of electrical charge. Any resulting net movement of positive or negative charge will generate a membrane potential or voltage across the membrane.
When the membrane of an axon is at the resting potential, most voltage-gated sodium channels are closed. When a stimulus depolarizes the membrane, some gated sodium channels open, allowing more Na plus to diffuse into the cell. The Na plus inflow causes further depolarization, which opens still more gated sodium channels, allowing even more Na plus to diffuse into the cell. Once the threshold is crossed, the positive feedback cycle rapidly brings the membrane potential close to E and A. This stage of action potential is called the rising phase. Two events prevent the membrane potential from actually reaching E and A. Voltage-gated sodium channels inactivate soon after opening, halting Na plus inflow, and most voltage-gated potassium channels open, causing a rapid outflow of K plus. Both events quickly bring the membrane potential back towards EK. This stage is called the falling phase. In the final phase of action potential called the undershoot, the membrane permeability to K plus is higher than at rest, so the membrane potential is closer to EK than it is at resting potential.
The first step in action potential arrives depolarizing the presynaptic membrane. On the second step, the depolarization opens voltage-gated channels, triggering an influx of Ca2+. The elevated C2+ concentration causes synaptic vessels to fuse with the presynaptic membrane, releasing neurotransmitter into the synaptic cleft. The neurotransmitter binds to ligand-gated ion channels in the postsynaptic membrane. In this example, binding triggers opening, allowing Na+ and K+ to diffuse through.